Hello everyone, welcome to a new technical tutorial from Team SMS. In this tutorial we will be learning how to quantify the phases present in a sample by the Rietveld Refinement method. For this purpose we will be using a software called as TOPAS. TOPAS stands for Total Pattern Analysis Solutions. TOPAS is a profile fitting based software for quantitative phase analysis, microstructure analysis and crystal structure analysis. In order to quantify the phases present, we will be needing two things. The first is the XRD raw file of the sample, and second is the all the CIF files of the number of phases present in the sample. For each of the phases we will be needing the CIF file in order to quantify. So let's begin. First of all, we have to load the XRD raw file. This is our XRD raw file. So then we need to click on the name of the raw file. Let's understand the interface a bit. This is called the parameter window. This is the XRD scan window. This is the kernel output window, and this is the fit window. We will be getting to it one by one. First of all, we have to click on the small triangle on the left of the name of the sample. First, we have to load the emission profile for this sample. So right click, load emission profile. It is kept in the desktop. So the machine on which this XRD has been carried out is a D8 advanced diffractometer, and the target use was cobalt. So the emission profile is a .lam extension file. So we will be using the first file. Then next, background. CIF polynomial should be ticked, and we have to refine this one. The order of the CIF polynomial can be around 4, 5. 4 or 5 will be a good fit. This is the background, refine. Next, we have to load the instrument parameters. Right click, load instrument details. Here it will be a .par file, and as I had said earlier that we had used a D8 advanced diffractometer with cobalt target, so we will be selecting this file. Yeah. Here we will be making some settings. This is the primary radius and this is the secondary radius of the goniometer. If you are using a point scan method, then you can use this point detector, but in our case we have used the linear XRD, so we will be unchecking this one and we will be clicking on the linear position sensitive detector, linear PSD. For this we have to set the primary solar and the secondary solar values. For our instrument it was 2.5. and we have to refine these values. Yeah. Here in the corrections we have to check this zero error, sample displacement and refine. Those are already set to refine by default. LP factor is the Lorentz polarization factor, and for unpolarized radiation the Lorentz polarization factor is 90 degree, that is for the X-ray diffractometer without any monochromator, and it is 0 for fully polarized radiation, that is synchrotron radiation. So as we had used an XRD diffractometer, so for our case this value will be 90. and we can refine this one. Next, we have to add the CIF files of the phases present in the sample. So from the XRD, we know that we have got two phases present in the sample, that is the austenite phase and the ferrite phase. So we will be adding two CIF files on this. So right click on the name of the sample and load CIF. First of all, we will be adding the CIF file of the austenite phase. Here, let's rename this one austenite. Okay. From here, we by clicking on this austenite, we can see the FCC structure of the CIF. So let's click on view or hide structure. So from here you can see, you can scroll, you can drag, and you can see that this is a FCC structure. So we have ensured that this is really an FCC structure. So from here we can see it. There are different settings available for viewing this one. Either you can remove these cell wall, this unit cell walls, you can remove these bars, you can decrease the size of these walls. So these all settings are available there, you can hover over it. Now we have to do some settings for this CIF file in order to fit the XRD profile. So here this is the lattice parameter. Here we need to refine and scale, refine. If you want to calculate the lattice strain and the crystallite size, here also you can refine. Either Lorentzian or Gaussian, anyone you can choose, and accordingly later we can fit it. So now let's run. Let's run the fit. Now here you can see there are two patterns. One is the observed or the experimental pattern, which is on the top, and the second is the calculated or the lower one is the difference between the observed and the calculated. So this is the difference. Our main aim of running this quantification is to minimize the difference curve. So now let's add the second CIF file. The other phase which we had is the ferrite phase. So let's add this CIF. As we have added, let's rename this one ferrite. Now again let's check, let's see the structure of this ferrite. Click on view or hide structure. So here we can clearly see that it is a BCC structure. You can drag around and you can check it's a BCC structure. So we have the correct CIF. These CIFs can be obtained from the ICDT database. Yeah, so let's change the settings of this ferrite CIF. Click and refine. Now let's run the fit again. Here in the output window, here in the fit window, you can see some parameters. Those are the RWP. RWP is the weighted profile R factor, and R factor, these are the Rietveld error indices, and in order to get a good fit, we need to minimize these indices. So our aim has to be minimize this REXP, and from here we can check the goodness of fit. Click on the name of the sample, and from here the second tab, RWPs. Here we can get all the data, R exponential, RWP, RP. These are all the Rietveld error indices which we need to minimize, and GOF is the goodness of fit. A goodness of fit of less than 1.5 would be a very good fit. So our purpose is to decrease the GOF so that the observed and the calculated, they superimpose on each other. So now, in order to superimpose, or in order to fit the observed and the calculated, here we need to do some more settings. Here we need to fit the lower one on the upper, or we need to minimize the difference. When moving the mouse onto the phase name, here as you can see, these are the phases present, austenite and ferrite. So prima facie we can see that it is showing 51% and the ferrite is 49%, 48.2. But here as you can see that the difference curve is used. That means it's not a good fit. So visually, first of all, we have to fit this one properly. Now a small observation here, that as you can see that when you are clicking on the name of the austenite phase, you are getting a yellow highlight on this, and here you are getting a blue highlight. As you can see, it is written in white and it is in blue. So when you are clicking on the austenite, you see that these are the phases which are getting highlighted in white. This one, as you can see, these are the austenite phases present, which has not been fitted properly, and will be fitting them now. And this is one austenite peak, this is another austenite peak, this is another austenite peak, and these are the other FCC peaks, 1, 2, 3. So we need to minimize the difference between the observed and the calculated. So here what we can do is, for the austenite, let's click on the preferred orientation. So here direction 1 and direction 2. In the austenite, the preferred orientation is this peak, and the second preferred orientation is this one. So we need to write down the HKL values of the direction 1 and direction 2. These are two are the preferred orientation in order to fit the curve properly. When we move the mouse onto the phase name, you see the intensity distribution of this particular phase will be shown using a bold line. Additionally, the HKL markers for this phase are highlighted using small triangles at the bottom of the markers. You can see here, when I am highlighting the austenite phase, you see these are the markers which are highlighting the austenite phase. So now we can see here that this is the 220, the 220 phase, and the first one is the 111 phase. So the first preferred direction is 220 and the second one is 111. Now let's run the fit. Still it has not fitted properly. So now let's run the preferred orientation for the ferrite CIF. For the ferrite, these are the preferred orientation. So first we can see that this is the direction 1 and this is the direction 2. So let's try to fit this one. Let's see what is the HKL of this one. It is 220. It is 220. Sorry, I'm sorry. This is 211 and this is 200. 211 and 200. So now let's run the fit. Yeah. So now you can see that we have got almost a very good fit of all the phases here, either the austenite or the ferrite. We have got a very good fit. Let's check the goodness of fit now. Yeah, so we have got a very good goodness of fit of around 1.26. A goodness of fit of 1 would mean that it is exactly superimposing the calculated and the observed are completely superimposing, but still we got a very good goodness of fit of 1.26, and as you can see that we got a very low value of RWP. Thus these are the final results, and visually also we can see that the difference has got minimized when we after fitting both the CIF files. So these are the final values. Austenite is 56% and ferrite is 48%. So in order to export this data, we have to right click here, right click, edit or print chart options, here copy to clipboard as bitmap, open a Word file. Here we can simply paste the data from there. Well, it did not copy it. Let's copy it again. Let's paste. Yeah. So here we can see that we got the fitted curve here. Now if we want to export the data, the GeoF data and everything, here we can go to click here and then click on the RPT. From here either you can click Control A and then you can copy and then you can paste here. Yes. So from here you can get a detailed, yeah, from here you can get a detailed value of the REXP, RWP and all the quantitative phase analysis. The quantity of the phase 1, that the austenite, here you can see 56.244% and the phase 2 ferrite 43.756%, and rest all parameters you can see here. The sample length, and then the corrections which we have applied, and the R brag values, and the cell volume, and the crystallite size, and the strain of each phase you can copy here from the data which we have obtained. So this ends the video, and in the description section we have shared some of the links from which you can get to the theoretical part of the Rietveld refinement method. Thank you very much.